Today we're going to be looking at D2.1, which is going to be the cell cycle, mitosis, meiosis, and the like. So let's start off with the broad picture, which is the cell cycle. So, what is a cell cycle? Well, it can be divided into multiple phases in a quick little ring. So, we're going to start off up here with growth 1 or the G1 phase. G1. Now, this is actually the longest phase by far, and it's where the cell replicates and grows because it just needs to continue its metabolic activity. Next is a synthesis phase, the S phase. Then we have the second growth phase, which is about here. And then finally we have the this mitosis. The M phase, that's the mitosis and stuff. And this is G2. So this is the second growth. This is the first growth. So that's G here. Now, in this phase, we also have cytokinesis, so /C. And each of these phases are defined by different things. So often you can either start at this phase or at either G1 phase or the mitosis phase, which is cell replication. Cytokinesis is the splitting of the cell itself, not just the nucleus, to create two identical daughter cells in most cases, other than oogenesis and in some plants forming runners or budding. So yeast especially sometimes can form little unequal cells because of cytokinesis, but that's not what we're going to be looking at right now. So how does the body actually decide when the cells, or when each of these phases are executed? So for that, we're actually gonna draw out a little graph. So the graph should look somewhat like this. And just to mark this. So this is gonna be on this axis we're gonna have the actual time, let's say, and on this axis we're just gonna have, well, nothing really. This is just a representation. So I'm gonna start off by just drawing a general broad curve, which is called the D. It's just cyclin letter D. So cyclins, let's just cut this. Cyclins control cell cycle. So cyclin, cycle, cell cycle. So you can just remember cycles control the cell cycle. It's all just C's. Now, from here on, you have the D cyclin, which kind of increases throughout and just guides all the other cyclins. Then you're gonna have the first major cyclin that we see, which is the E cyclin, which is gonna peak right about here. From there, let's label that as E. From there, we're gonna have the maybe about here. Gonna have the A cyclin, and finally, we're gonna have the B cyclin. Now, this is just a rough graph, but in theory, each of these cyclins correlate with a specific phase. So, starting at this phase, up to about midpoint, so about here, we have the G1 or growth phase. After that, the body naturally moves on, starting at that point, when that peaks, into the synthesis phase. Once synthesis is complete, at that peak, again at the A peak, we get the G2 phase. Finally, at the peak of B, we get, let's put that here, the mitosis. Now, this is a very general representation. There is a bit of tweaking that could be done. The cyclones don't all have such clear peaks, and there are some mild variations, but this is just how you can interpret it. So what cyclones do, as we said here, they control the cell cycle. Now let's move on to actually focus on like different parts of the cell cycle. So this, the G2 all the way till G1, excluding mitosis and cytokinesis, is actually going to be called the interphase. Okay, so the interphase, that's going to be where the cell spends most of its time, and we'll get into that later. But let's actually start with mitosis and cytokinesis, because that's actually how cells multiply. That's the most important thing in this case. So let's just take this down for a second, and then we're gonna raise this. Focus on mitosis. Beautiful. All right. So what is mitosis? Well, to put simply, it's the nuclear, I don't think that's spelling, nuclear division within a cell to produce two identical daughter cells. Now, how would that look? Well, say you have the parent cell looking like that. It would actually split and produce two daughter cells that look exactly like that. Obviously, we're using the word exactly a bit generously here because my shading is a bit off, but that is the main idea behind it. Now, how does this happen? There's a set of steps that take place to actually achieve this. So let's first just start with defining or listing out these phases. So you get the prophase, we then have the metaphase, then we have the anaphase, finally we have the telophase. Now, each phase is very characteristic, and you can actually identify them through a microscope, right? So in the prophase, we're going to have the chromosomes actually condense. So we actually, chromosomes condense. So we can actually start to see the chromosomes, and that's done by the chromosomes get supercoiled and we start to see them, but I'll cover it slightly later on. Now, in this phase, we also get the cell begin to split, but again, we're going to cover that in cytokinesis like at the same time. So this is the main phase. So the chromosomes start to condense, and we have a large nucleus that's already become large because of growth too. So we have a pretty large nucleus, and we start to see chromosomes. So let's say those are chromosomes, right? That's what you're going to get in prophase, right? So that. Next, we're going to go on to metaphase, which is actually quite interesting because the cells, the chromosomes start to line up at the equator. So line up at equator, Now that basically means that the cell itself begins to form the nucleus itself begins to form a equator. So that's a nucleus, begins to form a middle equator. And the chromosomes decide to line themselves up maybe not like that. Like this would be a better representation at that equator, right? And from here, we also start to see, it starts in this step, but it's quite visible, very apparent in the next, during metaphase, is that the centrosomes, these little things, begin to go to each pole and actually reach the pole. So you can call this pole, I don't know, left pole and the right pole. So they reach the poles, and then once they have done that, they actually start sending out these little fibers called microfibril tubules. So both of them start to send those out in metaphase, and it starts to line up. Once we have that, we actually move on to the anaphase. Now, what is the anaphase? That's where kinetochores, so let's go back to that drawing. I don't know why I erased that. Let's go back here. So, go. At this point, we have centrosomes. So I'm going to actually label these with different colors in a second. So, somes, centrosome. And this is actually a centromere. So the centromere is attached to the chromosome itself, while the centrosome is attached to the, well, not attached, but it's attracted to the poles. And these microfibril tubules actually attach themselves to the centromeres, as you can see here, and begin pulling the chromosomes towards the poles. The pulling is important because it actually separates the nucleus into two halves. So like you can kind of just visualize how the nucleus starts to split into two distinct sides so that we can actually make a new cell. Now, once we are at this step, once the things are being pulled, we move on to telophase. So anaphase is just the pulling. Ooh, let's go here. Pulling chromosomes to poles. And that's achieved. Let me just write this little highlighted thing. Kinetochores. So little kinetochores cores are going to be the ones who are doing most of the work. They attach to the centrosomes and they pull until they reach the core. Now at telophase, for this we're going to have a little less of a diagram, more of just an explanation, but we can still draw a little diagram. Everything's already reached where it's meant to be, so I'm gonna split this in half just to kind of look at it. We're gonna have the green side, which is so these are the little chromosomes. So the chromosomes have already reached their poles. The nucleus is visibly separated, like it's not separated yet, but you can tell there's a clear divide. And at this point, it's just the final stage. It's going to start, cytokinesis is going to start pulling this this nucleus apart with the rest of the cell, with the cell in like the cell fluid and stuff like that. So that's actually, so this is basically mitosis very quickly. So prophase, which is like this, we can see it all condensing. Then you go on to metaphase, which is where it lines up at the equator. Then we go on to anaphase, where it just pulls apart, and then finally telophase, where you can tell that there's two new cell membranes that are starting to form. And what I hadn't mentioned that I probably should mention is at this point, so nuclear membrane breaks, and then here distinguishes again, nuclear membrane reforms. Let's actually strike that, call it envelope. envelope, that's the actual term. So the nuclear envelopes break and reform. That's how you kind of know what's happening and when it's happening. It's kind of hard to see on the microscope because it's a really thin layer, but it is a key step. So that's how it works. So this is very brief definition. If you actually write this in your exam, if you just do dot points, you would get most of them, if not all the marks, with a little bit of explanation. So it should be fine. So let's erase this, take a second, and move on to actual cytokinesis happening simultaneously, so we actually understand this. So cytokinesis is a bit of a more drawn thing. So I'm just gonna erase that quick and go cytokinesis. Oops. Cytokinesis. Okay, so kinesis means to move. Cyto is like the cytoplasm, like this, the cytoplasm literally, which is the cell fluid. So I'm gonna start off with a cell drawn, say, in green, and within that cell we're gonna have the nucleus, right? That's a bit larger than you normally would have, but still. So this cell is about to start mitosis. Now we're gonna add a little bit of embellishments. Boom, everything gets pulled apart. So you're gonna have your chromosomes going to each side. Okay, so you have two pretty much identical halves now. Where do we go from here? Well, at this point, interesting little thing happens. A contracyl protein ring forms. So let's write that down here. I'll have that in red. Contracyl protein ring. Okay, so what's a contracyl protein ring? It's a pretty long, convoluted word. Basically, it's a ring. It's going to stretch, or actually go around the center, or the approximate midway point. So let's do a dotted line here, and then just lovely. Okay, so what does this contracyl protein ring start to do? Well, firstly, we don't need to know, but it's good to know. It's made up of actin and myosin. So these are the proteins that it's formed from. So actin and myosin. Yeah. So a contracyl protein ring, CPR, forms around and begins to pretty much squeeze the life out of, like, squeeze the center of the cell. It just starts to, if you have a ball, it's just gonna start like this. So we have a cell, the ring pushes, eventually gets really tight, and it can't really sustain, like, it's just pushing so hard, and you get two new cells. So you go from this to let's represent this with a new color. So you go from that to two completed new cells, as you can tell, each with the same amount of chromosomes, and those will decondense, and then you have growth 1, then synthesis, then growth 2, and then again you go back to mitosis and then cytokinesis. So this is the cycle, and this is the completed final product. So you started off with one cell that looked the exact same. You now have two cells that look the exact same, and this process does require energy, quite a lot of it actually. This happens so many millions of times every day. So growth requires this, fixing damaged tissues requires this. Happens a lot when you sleep because your body isn't really doing too much else. All right, so what else would we be focusing on in this thing, in this scenario? So we would obviously start to talk about the interphase. So the interphase will be covered in the next video.